right? Okay, so thank you very much for allowing me to give this talk. I think it's an area that is of great interest to me. I've been at McMaster for about 30 years, and I work mainly on E. coli and studying gene regulation. And my lab performed some of the first transcriptomic studies on E. coli in the early 2000s, late 1900s, latter part of the last century. And it was really an eye-opening experience because transcriptomics provides so much data that we really have to use the tools of bioinformatics to conduct robust analyses. And that's becoming increasingly common in many different fields. And that's really what I want to explore today. You know, we need bioinformatic analyses of large data sets. And why is this the case? And it's probably a big driver of this is that the cost of sequencing has dropped enormously. And if you look at the sequencing, you know, in 2005 or so, it cost $1 million to sequence a megabase of DNA sequence. And now it costs about 10 cents. And this is meaningful to me because we were sequencing a gene that was about 100,000 bases of length gene, and it cost us $100,000 to do it, which is exactly around this point. And now it would cost about a penny, according to this chart. So that dramatic drop in sequencing cost allows us to obtain just enormous amounts of data. And it makes a lot of projects feasible that prior to this cheap sequencing just weren't feasible at all. It would have been prohibitively expensive to do these experiments. And so I'm going to talk today not about transcriptomics or our basic studies on E. coli so much, but an area that I think has been of interest to me in the last 10 years, and that is DNA-based water diagnostics. And I want to talk about three things: microbial source tracking, and these are going to be brief topics, but I have a few slides to go through. A new DNA-based strategy for determining water quality that we're exploring here at McMaster. And then finally, I want to give a plug for our MacWater Diagnostic Research Initiative, which is an interdisciplinary initiative that brings together engineers, biologists, biochemists, and software developers. So with E. coli, you know, with water quality, we're always interested in how good is the water. And you know, a primary indicator of water quality is the presence of E. coli. And the trouble with this is it's not a great predictor of water quality in itself because most strains aren't pathogenic, right? So it's not, doesn't really tell us much about the water safety. A lot of us, we all have E. coli in our intestines in high amounts. I think it forms about 5% of the intestinal microflora. So that's one reason it's not a good measure. E. coli is just one of many. There's 50 different potential pathogens that can be present in water, and so E. coli in itself is not a really robust indicator. It's something that we can easily score, and it's often associated with fecal contamination, but it has lots of limitations. And you know, in recreational waters, high E. coli counts are associated usually with high rain events. And so they lead to misclassification of beach safety. Over the past 10 years, we've been examining beach safety in many areas, including Niagara region. And so with the Niagara Public Health Association and the Conservation Association, we sampled many beaches that for water quality that are the local health officials were interested in. And we did this for several years. And we've expanded these kind of partnerships to include the town of Waynflete, the city of Toronto, and other jurisdictions. And now we're part of a large group proposal to do this kind of analysis all over Ontario. So bioinformatics can be used a lot in diagnostics for different applications. And I've listed some here, and these are all important. They're all very important. There's diversity analysis, being able to sequence the genomes of potential contaminants. But today I want to talk about conserved sequence, DNA sequence indel analysis. And this is the idea that we can use specific sequences that are found only in groups of interest that can be used to identify and quantify unknown organisms. And this, we think, is a really promising strategy for estimating microbial community diversity, for identifying specific organisms. And the advantages it has over other organisms is that it's very highly specific for target organisms. It has high predictive power, and it has the ability to detect the presence or absence of groups of organisms in metagenomic sequences that are uploaded to databases. And so these can be used to identify pathogens, quantify them, and identify other microorganisms as well. So if we think of DNA as being this big ball of string here in a sample, traditional microbial diversity analysis relies on 16S RNA, a gene which gives us some information, but the sequence is all the same. And it has slight differences in sequence that can be used to classify organisms, but it gives funny, fuzzy data. So it's, requires complex analysis using large data sets. And the results are variable, depending on the type of analyses, the person doing it, the parameters they set. And so this is obviously something we want to address. We want something more specific. And conserved signature sequences are as an alternative, are present in all organisms of interest. They're different from one another. And this means we can use simple identification and enumeration just using a small number of sequences. We don't have to do complex matching algorithms. And it gives a single unique result that's independent of the user or the algorithm. These sequences, you count them, they're there or they're not there. So that's great. And this is an example of an indel. And I just want to give these as an indication of an indel in this case in a iron uptake protein, and this was identified by Dr. Rad Gupta's lab. And you can see that it's specific. This is shared by all the organisms in from X1 to Z2 and is absent in these organisms. And it can be mapped to a phylogenetic branching point. So this is the iron uptake protein. A student in my lab developed a specific test for this. And this can be used to differentiate E. coli, pathogenic E. coli 0157H7 from all other pathogens. And that's what's shown here. This shows the indel. This is specific in insertion. And these are positive controls for another gene. And this shows that this sequence is absent in related bacteria. So this can form the basis for a very specific PCR test. And we have thousands of such sequences that can be used to classify all kinds of organisms. But this was one of the few proof of principle studies that we did in the last few years. So environmental samples contain lots of information in the form of DNA. The complexity of DNA in a single water sample can be equivalent to several human genomes. And this is roughly the breakdown. We can identify viruses, bacteria, and free DNA, and we've published this information for the Great Lakes. And for water safety, we have to be concerned with several key contaminants shown in this slide, and I'll just go through it. But if we're going to identify DNA diagnostic sequences, we have to have a way of using this information to create new pathogen detection devices. And I've partnered with engineers to do this here at McMaster. We're creating specific devices for rapid detection of DNA. And I think this illustrates that, when we have a lot of bioinformatic information that yields something that's tangible or useful, we have to partner with people. And that means that you as students are going to have to be far more interdisciplinary in your outlook than, say, I did when I was your age. There we really didn't have bioinformatics in the sense that we do today. So the current water monitoring, if we apply this, is not very fast. It's not isn't robust enough. It's not comprehensive enough, and it costs a lot. So came up with this scheme, and which we call an integrated approach to water monitoring, where we take samples, we combine it with public databases, we're designing microfluidic devices that can measure biological parameters as well as chemical and physical parameters. We have an information fusion expert who integrates this information, combines it with weather data that can be integrated and returned to the end user. And so this is the scheme that we're working on, and we've been using this pattern for about 10 years. And it's really the feasibility of this is driven by several changes in technology. One I've talked about, inexpensive DNA sequencing, and the others are cheap and readily available semiconductor-based sensors and new modes of wireless transmission and analysis. So while these aren't, strictly speaking, bioinformatics, they are important enablers of the use of this technology. So our goal, and this is our mission statement, develop low-cost, real-time portable devices that can analyze environmental samples for these contaminants, and this is what is needed, and lots of people are working on this. We have a planned integration of our team, including engineers, that I think is unique, and that's what's shown in this slide, and shows all the people who constituted our original team. We have lots of external partners, and this is an outdated list. We have lots more now, many additional companies, because now I have companies calling me asking us to work on problems, and that didn't happen the first 20 years I was at McMaster, but in the last few years, I'm not surprised to get these kind of calls. So this is a great kind of integrated approach, and it readily lends itself to training, and this just shows some of the training objectives that we've had, and we think these complement some of our academic programs really well. We have a website, the MacWater Diagnostic Research Initiative, that I've just updated actually a little bit. So that tells you what we do, and we basically have 3 teams. We have surveillance, sensor systems, and information fusion. And I just want to thank some of the people that were involved, including Dr. Rad Gupta, whose work in this area of conserved signature indels and conserved signature proteins is really a made in McMaster technology that has been comprehensive, and it's been really wonderful to watch his career over the past 20 years as he just developed this field. He's published practically all the papers that are used to reclassify organism and to develop diagnostics. Tom Edge, working at Environment Canada, and a whole bunch of students. And of course, we're supported by funding agencies. This is a partial list. So I'd be happy to take any questions. I just wanted to mention, sometimes sampling doesn't go as planned. This is one of my students, and he's up almost to his bottom in a chicken manure pile. He kind of went through when he went sampling, but that's dedication for you. Okay, so I'm happy to take any questions. And I really want to thank you for giving me the opportunity to talk a little bit about our interdisciplinary team.
Thank you so much. Does anybody have any questions? Feel free to raise your hand. Magda?
Hi. Firstly, that was a really insightful presentation. I've never really thought about using tools to detect water quality. I was just wondering, what does your partnership with IBM entail? And can you speak a little bit more about that?
Well, with IBM as a partner in a lot of projects, and they contributed in-kind computing space because computing resources, and they were one of our early partners. We also have partners with waste treatment companies. Obviously IBM is a key resource for a lot of universities, and when we write a grant proposal, we obtain letters of support from industry, and IBM is a good example where they provide expertise. They themselves are interested in lots of things, and water analysis is one of them. They have a water initiative. And I met with their educational people. They are very interested in particularly logistics. We haven't really made that a big part of our program, but they told me that's something that IBM is really supporting. And it really struck me. I advise students, you should incorporate some training in logistics in your undergraduate experience, because really Logistics is a big thing now. You know, the movement of information or the delivery of goods from Amazon. Everybody gets Amazon packages every day. I mean, there's huge logistical issues. So that's they've been an important partner. We have other computer partners as well now, though. Any other questions? I just mentioned we have a monitoring project for Toronto beaches now. Some of you probably live in Toronto. So the Ministry of the Environment has funded a project between our lab and another to monitor Toronto beaches, to look at the use of a 4-hour enterococcus test to replace E. coli testing, because it provides more timely information. But we're also doing a comprehensive analysis of water pathogens in Toronto, because it really gets around some of the problems we have in identifying microorganisms. Some microorganisms are really hard to grow, like Campylobacter, for instance. You have to be an expert in Campylobacter culturing, and it takes about a week to get numbers. It's not like E. coli where you just grow them overnight. And yet, if you use something like a DNA-based technology, you can just extract the DNA and identify the numbers and types that are present in DNA, just as you would for any other bacteria or virus. And so I think this is an extremely promising approach. And just before this meeting, I was on a call with a new grant proposal that will do the same kind of work all over Ontario. So, yeah. Okay, I don't want to take up any more time. I think actually that's about where I was supposed to be. Yeah. Hi.
If I can ask
Tina,
a
how are you?
question.
Sure.
Hi, I'm good. I'm a first year, and I just wanted to know, what are some opportunities that undergrads could pursue to get involved and learn more about bioinformatics in your labs?
Well, you know, it's really challenging because there are a lot of undergraduates and there's hardly any professors. I mean, there are the same numbers of professor at McMaster as when I started here, about 950. I started in 1990, and yet now there's 3 times as many students. And so it's really challenging to offer lots of positions to everybody. And so I think to get involved, many students volunteer in labs. Try to take some lab courses. I know we have programs that don't have very many labs, but I think getting some lab experience is something you can do here. And when you leave McMaster, people will look at your transcript, and it has to make sense, you know? I mean, your transcript, you're going to be explaining why you took courses, why this all fits together. And I think we give a lot of latitude to students to take sort of whatever they want. And that's harder to explain. And when I was at your age, I took microbiology and I did a statistics minor at Guelph. And microbiology, we took lab courses all the time. And statistics also had about 10 required courses. So try to take tougher courses. You know, I know that's a challenge because everybody wants high grades, but you know, you really have to have a transcript that makes sense for you to move on to the step. For students who are in first year, it's difficult to really make suggestions. Just have really high grades. You know, work on keeping your grades up and try to get the occasional volunteer experience as a start. Okay, and then hopefully it'll be a paid position after that.
Thank you.